Hello, everyone. Thanks for sticking around, and thanks for the opportunity to talk. I'm a second-year neuroscience graduate student in Shefali Deste's lab. I've always been really interested in understanding mechanisms underlying atypical brain development. When we think about complex psychiatric illnesses and neurodevelopmental disorders like autism, we understand that it is very challenging to understand these syndromes basically because of the complex heterogeneity associated both at the genetic level and at the phenotypic level. So when we think about Dup15q syndrome, which has a well-known genetic etiology, it gives us this amazing opportunity to really try and understand specific clinical phenotypes that can be associated with the genetic variants. And I would like to think of the approach that we take in Shefali's lab as sort of this multi-level component phenomics approach. You already heard Rajitha and Charlotte talk about the motor and behavioral symptoms, and also some of the neurocognitive phenotypes. And we are also trying to collaborate with Dr. Peyman Golshani. You already heard him talk before, where we are really trying to understand what is happening in the Dup15q mouse models, and if we are really able to see the EEG signature that we're seeing in individuals with Dup15q, if we're able to see that in the mouse models as well. For the purpose of today's talk, I'm mainly going to concentrate on EEG biomarkers in our clinical population. And we've already identified that individuals with Dup15q syndrome have this significant increase in beta oscillations, which I'm going to show you guys in a minute. And before that, just to give a little bit of a background, I'm not going to give a lot of details. I know we have talked ample amount of times about the background of Dup15q syndrome. But I really want to highlight that why we're really interested in studying this syndrome is because of its high penetrance for autism and also intellectual disability, and they have a significant motor impairment and epilepsy. And when we think about this critical region here in Dup15q, we know that the deletion of this region results in Angelman syndrome or Prader-Willi syndrome based on whether it's maternally or paternally derived. And we also have duplications or triplications that actually result in interstitial or isodicentric duplications. So thinking about UB3A, we know that the hypothesis behind UB3A being the key gene that is underlying the mechanisms of Dup15q because of the symptoms that we actually see pronounced being in the maternally derived population. We also need to think about the other genes that are also present here. As you all know that there are about 40 genes present in this region, and UB3A has already been widely studied. We heard a lot of talks yesterday about UB3A, and we know it is very important in synapse formation and function. And also, there are other key genes that I want to highlight here are the GABA receptor genes. But something to keep in mind here is also that there are only 3 GABA receptor subunit genes, while there is around 16 GABA receptor subunit genes in total. And Dr. Sankar gave this beautiful talk this morning about how it's really important to keep in mind about the different subunits because they have different functionality. And I also want to highlight that there's a lot of research that are being done to really try and understand what these genes actually do. And also, we don't know how they function together and whether they modulate one another. Especially, I think Dr. Ben Philpott's group have already shown that UB3A does have a function in GABA co-release. And quickly to highlight again about the recent paper that Dr. Geschwin had published, they have shown that in Dup15q postmortem brains, there is overexpression in both UB3A and the GABA receptor genes in the Dup15q postmortem brains compared to non-syndromic ASD. This just tells us that the GABA receptor genes and the UB3A are critical and something to keep in mind as well. And I want to quickly go back to this paper that Shefali, this actually was published by Dr. Ryder's group. And Shefali was really interested and excited seeing this paper because this actually showed that in clinical EEG in individuals with Dup15q, they actually saw this background high-frequency oscillations that are present, which was very much like the EEG signature that you would see in patients who are on benzodiazepines. So this was really interesting because what we know about benzos are that benzos modulate GABA receptors, and that the GABA receptors are already present in this critical gene. We can imagine that there is a clear link between what is happening, the electrophysiological signature that you're seeing, and the GABA receptor genes. And so there are also papers that talk about when I think in mouse models, when they give benzos, they do see an increase in beta oscillations, and when they take away the drug, the beta oscillations go down. So this resulted in us thinking about these really important questions of whether we can actually quantify this EEG signature in this population, and whether or not the signature actually distinguishes our Dup15q population compared to those that do not have the syndrome, and also whether this EEG signature could actually relate to the clinical phenotypes like duplication type, age, and epilepsy. So to answer the first question is where we published this paper. This came out a few months ago already, but for those who don't know, I just want to highlight that, so we did publish that we could, this high beta oscillation is actually a quantifiable biomarker that actually distinguishes individuals with Dup15q from those who do not have the syndrome. And I want to quickly go about how we went about the study. Is basically we had a cohort of Dup15q population where we collected resting state EEG. This is basically a couple of minutes of EEG recording while the kids actually watched bouncing bubbles on a screen. You can imagine that as sort of a screensaver, basically, without any stimuli. And we also collected EEG from age and IQ-matched non-syndromic KST population, and also typically developing group that are age-matched with our Dup15q population. So we looked at different regions of the brain. These are the regions of interest where we looked at electrodes which are on frontal, central, and parietal, and we do a high-density EEG with 129 channel electrodes. I also want to highlight that we do put in a lot of care to actually come up with best possible processing methods for our EEG signal, because we collect high-density EEG, which also has high impedance, so there's a lot of noise, and we do deal with these challenging population of kids. So what we do is we use EEG Lab, which is a MATLAB software tool that is great for our processing. And we filter the raw data, we remove the bad channels, and we do something called ICA, which is independent component analysis, which basically decomposes multivariate signals into independent signals. And then we plot something called on the topo plot there, which is basically a heat map of the EEG signal power that you would see in the participants. And so moving on to our second question, whether this distinguishes our population from those who do not have the syndrome. So this is a figure that also was published in the same paper. To quickly orient you to the graph, the x-axis here is basically frequencies, and the y-axis is power. And you can see that the Dup15q syndrome population, which is depicted by the orange color in the graph, has this significant bump in the beta power compared to the non-syndromic AST group and the TD group, which is the blue and the green. And on the right is all the topo plots, which shows the heat map, which shows that there is a significant increase in the beta power on the bifrontotemporal regions of the brain. Coming to the next question of whether this relates to clinical phenotypes, this also was a question that was raised by Dr. Philpott yesterday. And we looked at age and duplication type and epilepsy. We did not see any difference with age, and we did not see any difference with duplication type. You would imagine that there would be a higher beta power in our IDEX versus interstitial because of the one additional extra copy of the critical region overexpressed, but that's not the case. But we did see a relation between epilepsy and beta power, in that children who did not have epilepsy had higher beta power compared to those who did have epilepsy. So after this, we really now wanted to actually take a step back and think about what are sort of the underlying mechanisms behind the EEG signature that we're seeing in this population. So the first question is, what is the genetic mechanism underlying these oscillations? And to explain that, we just have a few possible hypotheses, and I want to, again, orient you to our GABA receptor figure here, where the GABA receptors have a specific binding site for benzodiazepines, and this is separate from the GABA binding site. And what happens when benzos bind to GABA receptors is they keep the channel longer, and thereby they increase the cortical inhibition, resulting in high beta oscillations. But what we see here is that in our Dup15q population, we see the signature, but these are individuals who do not take benzos. So we can take a clue from this benzodiazepine mechanism, thinking about some possible mechanisms, and one of which is maybe there is an increase in the GABA receptor gene, which would possibly result in an increase in the GABA receptor subunits, which would then, you can think that there is increase in the GABA receptors functionally, which would result in an increase in the GABA binding sites because there's just more receptors for the GABA to go and bind onto, which then increases the amount of time that the channel is open, thereby increasing the chance of more chloride ions coming in. So this results in prolonged hyperpolarization, which makes the cell difficult for it to depolarize, thereby resulting in reduced neuronal excitability, and therefore resulting in cortical inhibition and resulting in high-frequency oscillations. But this is just one possible mechanism, and you can think that it's possible that there is still increase in the GABA receptor subunit expression, but it's still possible that there's just a lot of subunits that are floating around, but not being able to actually form functional receptors, which is really important to understand, because if there are not enough functional receptors, then it is possible that there are still not enough binding sites for GABA, but then why do we see this high beta oscillations, right? So the other possible mechanism that we can think of is actually having an increase in UB3A expression, which is possible. And I want to highlight again Dr. Philpott's paper that I mentioned before, where he actually has showed that an increase in UB3A actually results in an increase in GABA neurotransmitter overexpression. So it's possible that the increase in UB3A results in an increase in GABA. So there's more GABA that actually goes and binds to the already available receptors. And this would again lead to a prolonged amount of time that the channels would be open, leading to hyperpolarization and therefore resulting in the same phenomena of cortical inhibition. So how can we study this in our models? We are collaborating with Dr. Payman, and he already gave his talk this morning. So you already know that we're trying to establish in one type of models, we have already looked at whether there are high beta oscillations in the Takumi mouse model, which is basically the mouse model where the whole critical region is overexpressed. And now moving forward, we want to see whether this is actually seen in the UB3A overexpressed mouse models. And also, like he mentioned, it would be important to also look at the maternally derived mouse models as well. And like he mentioned, we will look at local field potentials, and also once we do see the high beta oscillations, then we can really think about figuring out where these beta oscillations are generated from using these mouse models. And also think about GABA modulators that we can try and use in these preclinical models to really understand mechanisms. And in our clinical population, what we can also do is look at other genetic variants that are also in this critical region, one of which would be edge duplication, where we have an overexpression of just the flanking regions, which would be important to consider, where you don't have RUB3A or the GABA receptor genes. And other thing we can also do is actually look at GABA conductance. So if we think that there's an overexpression of the GABA receptors and there's more GABA coming in, then in clinical population using MR spectroscopy, you can also look at GABA conductance to see if that is what is going on. And you can also, I think it's also important to look at stratifying the individuals with whether, depending on the duplication type, based on whether they are isodicentric or interstitial, to really understand gene dosage. And so we know that there is overexpression, and we know that there's increased cortical inhibition in this population. So clearly there is an imbalance in excitation inhibition. So we've been really thinking about how can we measure this in our population. VEP, which is basically evoked potential due to a visual stimuli, like a flashing checkerboard, for example, is actually a great tool to study cortical inhibition. And there are a lot of research that have been done in studying in other genetic syndromes where people have already studied excitation inhibition imbalance. They have seen atypical VEP. So we want to look at VEP in our population and also ask these questions of whether or not it is atypical and whether this actually stratifies our kids with Dup15q. And further on, we want to see if this actually relates to our beta power signature. This is really important because when we think about therapeutics and we think about using therapeutics and actually bringing this excitation inhibition imbalance to actually bring back to balance, then this would be important to study cortical inhibition and also have as sort of a target engagement, basically. So that brings me to the next really important question of whether or not we're able to actually validate this beta signature in terms of thinking about that as a clinical biomarker. I know Shefali is going to talk about biomarker in her discussion that is coming along after my talk. But I just want to highlight that the questions that we're thinking about in our minds to really understand whether this beta signature that we're seeing in this population is a biomarker of disease is, one, whether this beta power is modulated by state, and two, whether it's stable over time, and three, whether this is actually scalable. So the first question is to be able to, so the first one is to find out whether beta is modulated by state. We've actually thought about, I think the biggest change in state that we can study is from wake to sleep. So we decided to actually look at sleep in Dup15q syndrome population. So we have actually started collaborating with Dr. Jason Lerner, who is one of the authors in this paper, who worked with Dr. Shefali. And in this paper, what they found was in clinical EEGs of children with Dup15q, they actually saw this increased beta oscillations in the background EEG. And there's also parent report questionnaires that we collect in our lab, which is child sleep health questionnaires, which also talk about sleep problems in kids with Dup15q. And we also heard some of the parents talk yesterday about sleep problems. So we really were interested in trying to understand more about this and really seeing whether we are, in fact, seeing the beta oscillation persisting during sleep. So we collected clinical EEG data from Dr. Jason Lerner, who is the neurophysiology director core in UCLA clinics. And what he did was take overnight clinical EEGs from Dup15q participants, and he actually clipped the data into wake and sleep state data of 10 minutes each. And so in each recording, the wake and sleep is basically, wake is just the data collected when they were just before going into sleep. And sleep is basically when they looked for K complexes and presence of spindles. And in this figure, this is just two examples of our participants. These two are interstitial duplication patients. And you can see that the green represents the wake state EEG, and the red represents sleep. And you will be able to see that in both these participants, there is an increased beta oscillation during wake. And in sleep, you do see that in one participant, the beta oscillation attenuates, and in the other, it just stays stable. So there's definitely variation, but this also tells us that there is definitely modulation with state. I'm also looking into more participants now to really understand, and also I'm interested in looking at characterizing, really characterizing sleep in this population. So we are working with Dr. Jason Lerner to understand and score their sleep architecture to understand their sleep better. The next question that we wanted to understand is whether this beta power is modulated by time. to look at whether this actually attenuates with age across development, or if it changes when the kids actually move on to develop epilepsy. So this is a scatter plot showing all our Dup15q participants. The blue represent those participants with no epilepsy, and the red ones are with epilepsy. And the diamonds are interstitial duplications, and the circles are IDEX. The green dotted lines and the line in the bottom represent the average beta power in non-syndromic AST and the typically developing group. And you can see that the average beta power of Dup15q syndrome individuals are clearly high. And you can see all our participants who actually developed epilepsy are in the lower beta range here. And so these are the participants who actually came back for a longitudinal visit. And these two participants that I've highlighted here actually developed epilepsy between their first and second visits. So what you can see here is, this is the longitudinal data, and you can see that those two participants that actually went on to develop epilepsy, their beta power actually went down. So there could be one of a few things happening here. We are only looking at relative beta power here. So when kids develop epilepsy, there are changes in their EEG, and we're only looking at relative beta power, which is possible that the background EEG actually changes the relative beta power signature, or it makes it lesser. And the other reason is also that maybe we know that there's cortical inhibition in this population, and maybe developing an epilepsy actually changes the balance in EI, and so that actually makes their beta power go low. So the other really big question that we've been thinking about is whether or not this measure is scalable. So this is a really important question because we want to understand whether just with using clinical EEGs, if we are able to see the same signature that we see in our research high-density EEG. This is important because when we move forward with clinical trials, we want to be able to quantify EEG in individuals with Dup15q, and also be able to actually utilize the clinical EEG data that we already have collected across Dup15q clinics across the country. So the great news is that we are able to measure and compare. It looks comparable. So the first row here on the top is all 4 different participants that we have seen in our lab. Those are high-density EEGs from 129 channel electrodes. And those are, again, collected when kids were watching the resting state like bubbles. And the bottom 2 rows are the same participants, clinical EEGs, which is from 21 channel electrodes. The middle row is wake, which is just before sleep, and the bottommost row is actually sleep. When you compare the top 2 rows, you can appreciate that the data that have been collected from our high-density EEG is very comparable to the 21-channel electrode of the clinical EEG. And in the bottom, in sleep, you can just, like I mentioned before, there are kids where beta power actually attenuates, and there are individuals where beta actually stays stable. So I would like to thank the Dup15q Alliance for funding our grant again, because this actually gives us the opportunity to move forward with our proposal, which is basically to pull in clinical EEG data from across Dup15q clinics across the country and actually look at this much larger Dup15q population and try and find out if we are able to quantify beta biomarker and also be able to understand correlation between beta and also developmental outcomes and cognition in this much larger population. And we're also interested in further comparing beta power in wake and sleep states from clinical EEG, and also try and find out if for those kids where the beta power actually attenuates, if that actually results in, or if it is because they have better cognitive abilities, like the difference in beta power between wake and sleep, if that relates to their cognitive abilities. I just wanted to show this. So this is one of the kids that we saw from our Dup15q family meetings. We had such an amazing time, and I want to highlight that we actually collected EEG from 35 participants, both interstitial and isodicentric, epilepsy and no epilepsy, and within the age group of 9 months to 41 years, which is amazing. And we're really looking forward to be busy the next few weeks processing data. With that, I just want to thank all the Dup15q families and children that contributed to this research, without whom this wouldn't be possible. And I would like to thank each and everyone in my lab for making it such an amazing environment to work. And I would like to thank Dr. Jason Lerner. We are collaborating with him for getting the clinical EEGs, and Dr. Payman and Ben, who we are collaborating with in doing the Dup15q mouse model work. Thank you.